Hello, everyone. Thank you for joining our webinar today. I'm Sneha Poleo, a technical support scientist here at ACD. Before we begin, I have a couple of announcements. First, the audience will be muted for the duration of the webinar. If you have a question, please use the chat function to the right of the WebEx window. My colleagues will address your questions in real time. In the event we have not addressed your question, we will follow up with you offline after the webinar. Second, the webinar is being recorded, so in the event you cannot stay for the whole duration of the webinar, we'll have the recording up on our website. With that being said, I would like to begin the webinar. Here is a quick overview of the topics I'll be covering today. First, I'll present an overview of the RNAscope technology, followed by our product portfolio. Today, we'll focus on our multiplex fluorescent assays and how to choose the right assay for your research. Next, we will spend most of the time discussing the assay workflow, troubleshooting tips, etc. And finally, end the presentation with data analyses and FAQs. What is RNAscope? RNAscope is a novel in situ hybridization technology with unique probe design that allows for simultaneous signal amplification and background suppression to achieve single molecule detection at a single cell level while preserving tissue morphology. The technology consists of three main parts: a unique target probe that ACD designs against your sequence of interest, a signal amplification system that generates a high signal to noise ratio, and lastly, visualization of single RNA molecules as dots. The assay allows for the spatial mapping of mRNA in cells and intact tissues, all of which can be visualized with either fluorescent or chromogenic labels. The assay can be performed on a wide variety of sample types, including FFPE tissues, fresh and fixed frozen tissues, and cultured cells. Lastly, the RNAscope assays are rapid. Currently, we have over 15,000 catalog probes available, and new probes can easily be generated in about 2 weeks. Our standard assays can be performed in either 1 or at the most 2 days. The growth and adoption of the RNAscope technology is best exemplified by the number of peer-reviewed publications. We had our first publication in 2011, and since then there have been over 1,900 peer-reviewed papers published using the RNAscope technology in numerous journals, including many top-tier ones, as you can see. And the RNAscope technology is highly relevant across multiple fields of research, including oncology, neuroscience, infectious diseases, and stem cells. Here is a brief overview of the RNAscope workflow. RNAscope assay is a slide-based assay and is intended for use on sample types similar to that used with IHC or IF. The assay starts with permeabilizing the sample with reagents provided in the pre-treatment kit. The pre-treatment allows unmasking of the target mRNAs, thus allowing the target probe to penetrate into the cell and hybridize the target RNA sequence. The target probe is a pool of oligos that are designed using our established bioinformatics algorithm, which allows for target specificity. A series of amplification cascades can then amplify the signal, and RNA molecules can be visualized under the microscope as punctate dots. The signal can also be quantified semi-quantitatively or quantitatively using image analysis software. The primary feature of RNAscope technology is the probe design. Depicted here are the oligonucleotide target-specific probes as ZZs. They have 2 unique regions linked by a spacer. Additionally, each one of these oligonucleotide sequences has been designed using an informatics algorithm which selects sequences that specifically bind to the target sequence of interest and not cross-hybridize with any other sequences. The bottom of the Z complements hybridize to the target transcript. For amplification to occur, 2 ZZs must hybridize to the target sequence right next to each other in tandem. Once this happens, it creates a 50 base pair target-specific binding site on the bottom of the ZZ pair. The top of the Z is the base for the amplification structure. When the two Zs hybridize, it creates a binding site upon which a preamplifier can bind, and the amplification tree can be built on top. A standard RNAscope probe for a target sequence of 1,000 bases or more consists of about 20 ZZ pairs pooled together that are designed to hybridize next to each other along a target region. This allows for a tremendous amount of amplification and signal potential. However, only a few ZZ pairs are needed to bind to the target RNA sequence to generate enough signal for molecular detection. Here is a schematic showing the hybridization and the subsequent amplification of the RNAscope assay. The double ZZ probe will bind to the target mRNA transcript. Once the double ZZ probe binds, it serves as a foundation and binding site for the preamplifier molecule. Once the preamplifier is bound, it serves as a binding site for the amplifier molecules. And each amplifier can further bind multiple labeled probes, sequentially hybridizing to assemble a branching complex at each ZZ binding site. Labeled oligoprobes may either be conjugated to an enzyme, such as HRP, that generates a visible signal after chromogenic reaction, such as with DAB, detectable under a standard bright field microscope, or generate a fluorescent signal, such as with TSA-based fluorescent dyes, detectable under a fluorescent microscope. The labeled probes can also be directly conjugated to fluorophores that allow for visualization of the signal, again under a fluorescent microscope. This signal amplification strategy provides for visualization of the target RNAs as a single dot, where each dot represents an individual RNA molecule. Now, if a single Z binds to the target mRNA transcript, the preamplifier cannot bind as it needs both Zs to provide a binding site. This helps to ensure the specificity of the probes and helps reduce potential background staining. In order to detect multiple mRNAs simultaneously, multiple amplification trees must be built. With that in mind, we must ensure specificity. The specificity is ensured by adding a unique tail sequence to the probes, meaning to the double Z probe design, we have a tail sequence that is indicative of a channel designator. For example, a C1 probe has a specific C1 tail sequence that will only allow for binding of the subsequent C1 preamplifier, amplifier, and labeled probe molecules. Likewise, a C2 probe will have a separate and unique tail sequence. This slide presents all the assays that are currently available from ACD. For example, if you are interested in a single RNA target, you may choose our singleplex assay, such as the chromogenic brown or red assay. If you have two targets of interest, you may choose the chromogenic duplex assay. Both of these assays are available on manual and automated platforms. However, if you're interested in multiplexing, our multiplex fluorescent assay allows to detect up to 4 targets simultaneously in a single tissue section. On the other hand, if the target of interest is shorter than 300 base pairs or is a circular RNA, we recommend the base scope assay. As noted in the previous slide, some of these assays are available on either the Leica Bond RX or Ventana Discovery Automated platforms. Please note our Ventana Universal HRP AP assays have replaced the Ventana 2.5 HD chromogenic assays. If you're interested in the automation platform, please contact your area account manager. We will have our local field application scientists available for training and troubleshooting. The rest of the webinar will focus on the RNAscope Multiplex Assays. In the next few slides, I'll discuss the RNAscope amplification and detection systems specifically for the different fluorescent assays. Presented here is the amplification system employed in our fluorescent version 1 assay. All the 3 probe channels have independent amplification trees, such that the C1, C2, and C3 probes will have their own amplification trees stemming from where the probe has bound the mRNA transcript. The signal can then be visualized from the 3 independent fluorescent label probes. Please note that our C1 probes by default are ready to use in 1x in concentration, while the C2 and C3 probes are provided as 50x stocks. In order to make a 3-plex probe mixture, combine each of the C2 and C3 probes with the C1 probe in 1:50 dilution. And in the event you would want to run the C2 or C3 probes alone, please use the RNAscope probe diluent and combine in 1:50 dilution. The fluorescent V2 assay, on the other hand, can be used to detect up to 4 mRNA targets. Here, 4 independent amplification trees are built such that the C1, C2, C3, and C4 probes will have their own amplification trees built from the mRNA target. The signal can then be visualized using PerkinElmer's, now Akoya, TSA-based amplification system, or using the Opal dice. Now we'll go over how do you choose the most appropriate multiplex assay for you. Which fluorescent assay would be right for you? Currently, we offer 2 types of multiplex fluorescent assays, one of which is also available on the Leica automation platform. RNAscope Fluorescent version 1 assay, which is our first generation assay, is an all-in-one kit ideal for fresh or fixed frozen tissues as well as cultured cells. This assay allows for detection of up to 3 mRNA targets simultaneously in your tissues or cells, while the version 2 assay is a TSA-based assay, ideal for FFPE tissues, tissues with high autofluorescence, low-expressing targets. It was developed originally with FFPE tissues in mind to help address the autofluorescence issue. Besides RNAscope reagents from ACD, it utilizes the PerkinElmer's pyramide signal amplification technology, which is TSA, for further signal amplification. V2 assay is capable of detecting up to 4 targets simultaneously using Opal dice. This assay can also be followed with IF. The Leica Automated Multiplex Fluorescent assay is similar to the manual V2 assay. It also utilizes the TSA amplification system, but is fully automated and is compatible with FFPE and fresh frozen samples. Next, we'll discuss the assay workflow. With RNAscope assays, you may perform the assay as a one-day assay, though there are a couple of options to split this protocol into 2 days. The first option would be to complete the sample preparation and pretreatment on day 1, and then begin with probe hybridization and detection the following day. Please do refer to the technical notes recommended for your sample type of interest to determine which stopping point may be the most feasible. If unsure, please do contact technical support. The second option for splitting into a 2-day assay would be to complete sample preparation, pretreatment, and probe hybridization on the first day, then incubate the slides overnight in 5x SSC, which is saline sodium citrate buffer. The following day, you would remove the slides from 5x SSC, wash with wash buffer, then follow with the amplification reagents. This is highly recommended, especially for the multiplex fluorescent V2 assay, as it is a longer assay with several more hybridization steps. Please note in-house we use the 20x SSC purchased from Fisher. Here's the workflow overview for our fluorescent version 1 assay. The total runtime is about 6 hours from sample pretreatment to counter stain and mounting. Please note that you are provided with 3 different AM4 options, but you only need to use one of them. You can choose one of the alt options, either A, B, or C. I will explain in the next slide how to choose one of these. Each AM4 alt option is a cocktail of label probes conjugated with fluorophores. For example, if you use AM4 alt A, your C1 probe will be visualized in Alexa 488. C2 will be visualized in ATO 550, and C3 will be in 647. Based on the alt used, you can have the C1, C2, or C3 probes labeled in Alexa 488, for example, here. The different alt options provide the customers the flexibility to mix and match the colors. Here is an example of successful staining using our 3-plex positive control probes with fluorescent version 1 assay on HeLa cell pellets. Here is a detailed workflow for the Multiplex Fluorescent V2 assay. Similar to the previous workflow diagram, pre-treatment is completed first, followed by the target probe hybridization and subsequent amplification steps. This differs from the previously mentioned fluorescent assay as each channel is developed sequentially. Please note this is an enzyme-based development, and the enzyme used here is HRP. The AMP1, AMP2, and AMP3 steps are a pooled combination of the pre-amplifier and amplifier molecules so that the amplification trees begin to form simultaneously. This schematic shown here uses the Opal 520, 570, and 690 in the C1, C2, and C3 respectively, but these may be swapped as per your preference. Please note that this is a TSA-based assay and requires the purchase of TSA plus fluorophores or Opal dyes from PerkinElmer, Malakoya. Regardless of however many targets you are detecting, all 3 AMP steps are required to amplify the signal. Skipping any of these AMP steps would result in no amplification and hence no signal. Since the total runtime is about 14 hours, we recommend stopping the assay after probe hybridization. And as mentioned before, please leave the slides in 5x SSC overnight at room temperature. To run the V2 assay, besides the probes and the reagents that come from ACD, you would also require the fluorophores mentioned before from PerkinElmer. Presented here are our recommended Opal dyes that will be required for full plexing. Again, please note fluorophores are not part of the detection kit, and make sure that these are available to you prior to running the assay. Here is an image showing detection of 4 targets using our version 2 assay. Getting started. Some general tips to ensure success with RNAscope and BaseScope assays. First, follow the protocols. We have various protocols that are complete with details to help address the best way to run the assay. Next, sample pretreatment is very important to ensure that you get the most optimal RNAscope signal. The pretreatment conditions will differ from sample type to sample type and may require some optimization. Using controls is highly recommended, especially when you are troubleshooting your assay. And lastly, it is critical to make sure that you have all the components required to run the assay before you begin the assay. This is a standard RNAscope kit configuration, which includes the necessary pretreatment reagents shown on the left, the detection kit in the middle, and the wash buffer. Not pictured here are the probes. The reagent kit comes with 3 kits, which includes the pretreatment reagents, the detection, as well as the wash buffer. RNAscope 4-plex ancillary kit is required if you're planning to detect 4 targets with the V2 assay. Wash buffer stocks may show some precipitation on storage. Please make sure to warm the 50x stock at 40 C for about 20 minutes or until the precipitation is gone. Presented here are some recommended accessories required for running the manual RNAscope and BaseScope assays. To the top left are the 2 options for washing and handling the slides. You may use either TissueTech, which is not shown here, or the wash tray, common in most labs, or the Easy Batch slide processing system, which was designed to streamline your RNAscope and Bayescope assays. It saves handling time when working with a large number of samples. So we highly recommend that you use our Easy Batch slide processing system. The hybridization system to the right is designed to maintain the optimal temperature and humidity to ensure optimal and consistent RNAscope and Bayescope staining. We do not recommend using your own hybridization systems for running RNAscope assays. Shown below are the two methods for carrying out the heat-mediated target retrieval steps, which are required for sample types such as FFPE and fixed frozen. Our website has training videos on different steps of the manual assay, which is a great visual guide to assist with running the RNAscope manual assay. Here's the checklist. On the checklist, there are some key components that will help ensure your success with RNAscope assay. For example, using SuperFrost Plus slides, using the HiBZ oven, using control probes to analyze your samples, and using appropriate mounting media. For example, for the fluorescent assays, we recommend the Prolonged Gold. We also have some recommended general guidelines. For example, once you begin the assay, do not let the slides dry out. Do not skip any of the amplification steps. Do not alter the protocol. Again, this information can be found in the user manual for your future reference. Besides preparing samples according to our recommended protocol, it is very important to have probes controls for the RNAscope assay. First, if you're running the RNAscope assay for the very first time, we recommend running positive and negative control probes on our control slides, such as HeLa, in order to ensure that the assay setup in the lab is optimal. Then proceed running the assay using our control probes on your sample to assess the RNA quality and to determine the best pretreatment conditions based on the signal detected and tissue morphology. If your sample passes QC, you can then move on to running your target probe. Otherwise, it is recommended to verify your technique, check RNA quality, or perform assay optimization. I'll be discussing our scoring guidelines in a later slide. With any new sample you're working with, we always recommend running our 3-plex positive and negative control probes to assess the sample RNA quality and the assay conditions. Here is an example of the expected positive control staining in each channel. Running the 3-plex control also allows to ensure that you are observing staining with all the fluorophores and that the imaging and filter settings are optimal for the fluorophores used. The negative control is critical in catching any tissue-based or assay-related background. We have species-specific 3-plex and 4-plex positive control probes available. The negative control probe has probes targeting a soil bacterium gene, DAPB, which is absent in the tissue and hence acts as a negative control. To detect your gene of interest, please check our catalog online. Currently, we have more than about 15,000 probes in our catalog. If you're working with a novel gene and no catalog probe is available, please feel free to contact your area account manager or go on our website to initiate the custom probe design feasibility test. Again, for the V2 assay, please ensure that you have the appropriate fluorophores before you begin the assay. In the next section, I will share some troubleshooting tips for the multiplex fluorescent assays. For the fluorescent version 1 assay, we recommend using fresh frozen samples. We recommend that the section thickness is 10 to 20 microns. Thicker sections tend to detach easily and may require optimization of tissue permeabilization. The C1 probe is ready to use, whereas the C2 and C3 probes can be in 50x concentrated form. And to make a 3-plex probe mixture, please mix the C2 and C3 probes in your C1 probe in 1:1:50 dilution. If the C2 and C3 probe channel, the probes that you're using, you want to use them alone, please use the probe diluent in place of the C1 probe. For best signal detection and preservation, make sure that you use the prolonged gold mounting media. The version 2 assay is compatible with all sample types and especially is recommended for FFPE samples and if your targets are low expressing. It is capable of detecting up to 4 targets at the same time and can be combined with immunofluorescence. Please make sure to order either the TSA plus fluorophores or opal dyes from PerkinElmer before running the assay. Again, prolonged gold is our recommended mounting media. How to troubleshoot for autofluorescence? Due to the long fixation times, FFPE samples tend to have the most inherent autofluorescence, which poses challenges for image analyses. Our TSA-based version 2 assay helps to address this issue as adjusting the TSA concentration allows for a higher signal-to-noise ratio. We recommend that you assign high-expressing targets in the green channel and to use the multispectral imaging system or software for the background subtraction. The images here show how the protease treatment can affect the RNAscope signal quality, protease treatment being one of the pre-treatment steps. On the top, with the mouse kidney samples shown, you can see there is quite a bit of tissue autofluorescence. But when the sample is appropriately digested using Protease-4 at room temperature for 30 minutes instead of at 40 degrees Celsius, you can see distinct green and red dots. When the protease digestion is performed at 40, the tissue was over-digested, and you could see there was a loss of punctate signal and increase in the background, shown on the left, top left. Similarly, as shown in the bottom panels, with adequately treated fresh frozen mouse brain samples, there is no background on the right, and you can see beautiful punctate signal, but when the tissue was over-digested, there was an increase in background and reduction in signal. How do you troubleshoot for sample detachment FFPE samples? If you're working with FFPE samples and observing tissue detachment, we suggest that you add a baking step before the hydrogen peroxide, which is the first step of the pretreatment. And if you're observing tissue coming off from the slides after the protease treatment, add an additional baking step before that, meaning after the target retrieval. Both baking steps are for 30 minutes at 60 degrees Celsius. Troubleshooting sample detachment with fixed frozen samples. For sample detachment with fixed frozen samples, we recommend to dry the slides at -20 for 2 hours after cryosectioning. This step has been shown to be one of the most critical steps to make sure that the tissue does not detach. Additionally, we can add a post-fixation before the hydrogen peroxide for about 15 minutes to an hour. Additionally, the sample detachment also happens mostly during the target retrieval step, which is the boiling step. So we recommend adding an additional baking step before the protease step. And if you still observe sample detachment, then we recommend that you omit the target retrieval step and only do the protease digestion, but now extend the protease time to 45 minutes instead of 30. Please do contact support if you're still trying to figure out the best troubleshooting path for your issue. With the fresh frozen samples, we recommend to extend the fixation if you're observing tissue detachment, and you can extend to up to an hour. Additionally, we can add a baking step after the dehydration of the slides. Another common issue is seeing no or weak signal. To troubleshoot, please make sure to check the filter settings and image acquisition settings. Also, please ensure that you have performed all the AMP steps. Another possible root cause could be compromised sample RNA quality or suboptimal pretreatment conditions. We recommend to run the 3-plex positive control probes to assess the RNA quality and to follow the recommended pretreatment conditions for your specific sample type. Next, I'll share with you some published customer data. Here is an example where the authors developed an atlas of adult mouse spinal cord cell types and molecularly characterized 43 neuronal subpopulations that can be used to identify activated populations following behavior. So they used our RNAscope technology to validate co-expression of pairs of genes that belong to one cluster or population as identified from their RNA-seq data, as shown on the left. So if you see here on your left panel, they identify these two clusters, and what they're seeing on the right here are looking at these two clusters and staining them for these two specific genes of interest that were identified from the RNA-seq, and now they are looking at the staining pattern for these two pairs of genes and looking at the co-expression, and they could find co-expression of these genes as shown on these two panels here. Herring et al. In their paper here used RNAscope as a validation to validate the existence of different molecular subtypes of neurons identified by, again, their single-cell RNA-seq data in mouse spinal cord tissue. Moreover, analyses of neuronal subtypes in the context of the architecture of the dorsal horn of the spinal cord. And this enabled the creation of a spatial map where they could show the organization of neurons in multiple distinct layers. So they used RNAscope technology as a medium for in situ expression analyses of multiple genes. And what they could do here, they had, if we focus on 3 different GLUT clusters that they identified and named GLUT 5, 6, and 7, and we are looking on the right for gene expressions of these populations. And if we just focus on GLUT 5 and GLUT 6, we can see that the expression of this NMU gene, there is no expression of NMU in the GLUT 5 populations, whereas in the GLUT 6 population, there is NMU expression. So with RNAscope, they could actually unique expression patterns of these cells could be identified. One of the most asked questions is, can you combine the RNAscope-ish with IFC or IF? And the answer is yes. Both the procedures do have a similar workflow shown here that requires sample fixation, pretreatment to unmask your RNA or protein, and subsequent detection. For IF dual staining, we recommend running Multiplex version 2 assay. As compared to the version 1, the version 2 assay signal is more stable due to the TSA signal development, and it survives better after the subsequent IF procedure. Secondly, we recommend running RNAscope assay first, followed by IF. Next suggestion is to optimize the IF assay separately using the RNAscope pretreatment reagents to ensure your protein can still be detected following the RNAscope pretreatment. RNAscope pretreatment requires a protease digestion step to unmask the RNA. So do keep in mind as to optimize the IF. Lastly, the dual-IF assay works better for highly expressed proteins due to the protease treatment just mentioned, because the protease treatment can diminish the protein signal to some degree. And if you do notice the protein detection becoming weaker after combining with the RNAscope assay, you may try to increase the antibody concentration or reduce the protease treatment. We do have a detailed technical notes available on our website. If you have further questions regarding IF, please do contact technical support. This is an image again from Herring et al., and what we are seeing here is actually they have combined RNAscope with IF, where they have used GLUT1 antibody combined with their RNA detection. GLUT1 is seen in red. Next few slides, I'll go over understanding the RNAscope signal. We usually have customers asking, what does a dot mean? What is the significance of dot size? What is the difference between a dot and a cluster? With RNAscope assay, the mRNA molecules are detected as punctate dots, and each dot represents a single copy of mRNA. When multiple copies of your gene of interest are detected, the overlapping signal will form a cluster. So here, the yellow arrows point to single mRNA molecules with fewer ZZ probe pairs bound to them, compared with the dot pointed by the teal arrow, which is a bigger dot and still a single mRNA molecule. If you look at the clusters pointed by the white arrows, it is a cluster with an irregular rather than the round shape, resulting from overlapping signals from multiple mRNA molecules. RNAscope staining can be quantified by manually counting the number of dots per cell. It is also known as the semi-quantitative scoring method. With this method, a score of 0 to 4 is assigned based on the average number of dots per cell. Alternatively, for more quantitative approaches to data analyses of the RNAscope results, you may use the image analysis software. In-house, we use Halo software, which enables automated signal dot and cell detection, and provides output data in a format of average number of dots per cell or percentage positive cells. To learn more about RNAscope fluorescent image analyses, please feel free to check out the listed resources on our website. And if you would like to know more about Halo software, please contact Indica Labs directly. Additionally, if you're interested in learning more about image capture and analysis, please do attend our webinar next month, titled How to Correctly Interpret Your RNAscope Images, and it will be on March 26th at the same time, 9 a.m. PST. With that, I would like to thank everyone for attending today's webinar. My colleagues are still on the line to help address your questions, so please feel free to continue sending your questions through. Again, as a reminder, this webinar is recorded and will be available on the website. In the event you were unable to get a question sent through the chat feature, I'm leaving up the contact information for the technical support here. Please do stay online for a little longer, as we will be sharing some frequently asked questions with you shortly.